During interphase, the period during which the cell is not dividing, the chromatin is enclosed within a clearly defined nuclear envelope. Within the nucleus, one or more nucleoli are visible. Outside the nucleus, two centrosomes lie adjacent to one another, each containing a pair of centrioles. In prophase, three activities occur. First, the nucleoli disappear and the chromatin condenses into chromosomes. Second, the nuclear envelope breaks down. Third, the mitotic spindle is assembled. Also, microtubules from each centrosome connect to a specialized region in the centromere called a kinetochore. Metaphase begins when the chromosomes are distributed across the metaphase plate, a plane lying between the two poles of the spindle. Metaphase ends when the microtubules still attached to the kinetochores pull each chromosome apart from two chromatids. Each chromatid is complete with a centromere and a kinetochore. Once separated from its sister chromatid, each chromatid is called a Anaphase begins after the chromosomes are separated into chromatids. During anaphase, the microtubules connected to the chromatids, now chromosomes, shorten, effectively pulling the chromosomes to opposite poles. The microtubules shorten as tubulin units are uncoupled at their chromosome ends. At the end of anaphase, each pole has a complete set of chromosomes, the same number of chromosomes as the original cell. Telophase concludes the nuclear division. During this phase, a nuclear envelope develops around each pole, forming two nuclei. The chromosomes within each of these nuclei disperse into chromatin and the nucleoli reappear. Prophase 1 begins like prophase of mitosis. The nucleolus disappears, chromatin condenses into chromosomes, the nuclear envelope breaks down, and the spindle apparatus develops. Unlike mitosis, once the chromosomes are condensed, homologous chromosomes pair, a process called synapsis. These pairs of homologous chromosomes are variously referred to as tetrads. At metaphase 1, homologous pairs of chromosomes are spread across the metaphase plate. Microtubules extending from one pole are attached to the kinetochore of one member of each homologous pair. Microtubules from the other pole are connected to the second member of each homologous pair. Anaphase 1 begins when the homologous pair within tetrads uncouple as they are pulled to opposite poles. In telophase 1, the chromosomes have reached their respective poles and a nuclear membrane develops around them. Note that each pole will form a new nucleus that will have half the number of chromosomes, but each chromosome will contain two chromatids. Since daughter nuclei will have half the number of chromosomes, cell that eventually form will be haploid. In prophase 2, the nuclear envelope disappears and the spindle develops. There is no chiasmata and no crossing over of genetic material as in prophase 1. In metaphase 2, the chromosomes align singly on the metaphase plate, not in tetrads as in metaphase 1. Single alignment of chromosomes is exactly what happens in mitosis, except that now there is only half the number of chromosomes. Anaphase 2 begins as each chromosome is pulled apart from two chromatids by the microtubules of the spindle apparatus. The chromatids, now chromosomes, migrate to their respective poles. Again, this is exactly what happens in mitosis, except that now there is only half the number of chromosomes. In telophase 2, the nuclear envelope reappears at each pole and cytokinesis occurs. The end result of meiosis is 4 haploid cells. Each cell contains half the number of chromosomes, and each chromosome consists of only one chromatid. Later in interphase, a second chromatid in each chromosome is replicated, but the cell will still have only half the number of chromosomes.